we will move on to our next speaker, Magdalena Franchois. She's from the Community College of Denver, also Red Rocks Community College and Arapahoe Community College, to make sure that she gets all the classes that she really wants. And she is hoping to attend University of Colorado at Denver to study civil engineering this fall.
Great, hello. So this summer for my project, I was looking at an alternative way of detecting sulfide in wetland soils. I was working with my mentors, Laura Ree and Eve-Lyn Hinckley from CU Boulder. So I'd like to thank them for all of their help and support. I'd also like to thank the RECCS program for having me on and giving me this opportunity to do this research. Next slide, please. So sulfide is part of the sulfur cycle, which is a biogeochemical cycle that's easily manipulated by humans, but can also be affected via natural means. My method is intended to be used as a complement to traditional means of detecting sulfide to reduce the potential for error. My project focused on creating a standard operating procedure for use in alpine wetland field sites. Next slide. So first of all, what happens when sulfide comes in contact with silver film? So the reaction creates coloration that is dark gray to black in color, depending on the concentration of sulfide. So I am using this principle to use these films for detecting sulfide, right? So basically, if the film turns black or darkens in color, it means that there's sulfide present. So I'm exploring this method. It needed to be portable for use in the field. The field site it's intended to be used in is difficult to access. I needed to create a standard operating procedure for use in the lab. Next slide. So my project was mostly methods-based, nailing down this method. So after a lot of trial and error, and a few challenges that I came across and unknowns that I needed to deal with, I landed on mainly the equipment that you see in the top right and bottom left for use in the field. On the bottom right is a picture of my field site and the sites that I placed these samples in, the little mini core that you see in the top right. The top left is the setup for the concentration matrix that I created in the lab. Next slide. So this produced, so these methods produced these two matrices, right? So the one on the left is the one that I created in lab, which compares the concentration of sulfide to the amount of time that it's being exposed to the film. So you can see that exposure to sulfide results in coloration that has a rough correlation to concentration. There's also a time element here. You can kind of see as the concentration of sulfide increases, the amount of time that it's being exposed to the film seems to matter less and less. If there's a very low concentration of sulfide, then it's also hardly detectable no matter how much time it's being exposed to the film. And on the right is my wetland marsh study. So the marking P1 through P4, that was all different parts of my field site. And I put these films in for different amounts of time. So you can see that even at 5 minutes, there is a slight darkening in film. So at the very least, it's detecting sulfide, but it doesn't really say much about how concentrated the sulfide is in particular areas, which you can more clearly see the longer the film is exposed. So the bottom part with 18-hour and 24-hour sampling times, you can see what looks like gradient, sulfide gradient in the soil, parts where there may be higher sulfide concentrations versus lower. You can see parts where the film was exposed to plant matter and sediment because those areas aren't as deeply colored. So both of these demonstrate the link between exposure time and concentration. You know, there is an optimal time to expose these films based on the concentration of sulfide in the surrounding area. And then another interesting thing is with the wetland marsh study is it seems to suggest that sulfide reduction is happening continuously throughout these soils at all times, because the film only gets darker and darker, and there is a point where it goes beyond, it doesn't really get much dark, like it's not completely saturated or colored. Next slide. So my conclusions, this method can detect sulfide. So that's great. That can help. Sorry. So this is what I wanted. Like at the very least, it can detect sulfide, right? So that means that further analysis of these soils is justified. As long as the sulfide is present above a certain concentration. So in future work, I would like to explore the inconsistency with the reaction of sulfide with the film. I'd like to explore why this happens, because that was something that I noticed in my lab work. And it's also interesting to note that this method will be used in the near future by my mentor, Laura Ree. So that's very exciting. And that is my conclusion. That is the end.
All right, really great job, Magdalena. So we have a question here. Does the presence or absence of water saturation affect the sulfide measurements?
I'm not quite sure. You know, all of the tests that I performed was sulfide suspended in an aqueous solution. So I haven't tested any dry sulfide and its interaction with this soil. Because this method was, because I was exploring the use of this method in wetland soils, I didn't consider that.
Okay, thank you. Do we have any other questions out there? If you are on Facebook Live and have questions, you can post questions in the comments. We are monitoring those questions and we can pass those on to our panelists. Okay.